Today we're going to continue a series of lectures on the introduction to clinical trials. My name is DeMets, and I've been involved in clinical trials throughout most of my career through the view of a statistician and someone who's been interested in the science of clinical trial methodology. Over the past 4 lectures, we've covered a series of topics, beginning with sort of the background in the setting of clinical trials, why we do them in the first place. We also examined the challenge of defining the question. Often sounds simple, but in fact can be quite challenging to precisely define the question that we're going to be addressing. The last lecture, we examined carefully the basic designs used in clinical trials, ranging from the basic, simple, randomized parallel design trial to more complicated designs such as factorial designs and crossover designs. The key point from that lecture was that you need to select the design that matches the question that you're posing. So in today's lecture, we're going to examine another important part of a clinical trial design, and that is how large should the study be, the sample size estimation problem. There's a reference here which is a very nice, readable reference on the various aspects of sample size estimation that was written for clinicians and for non-statistician audience. The fundamental point that we want to stress is that every trial conducted must have sufficient statistical power or sensitivity to detect the differences of clinical interest. We'll define some of these terms in more detail later. Interesting enough, there's a lot of trials that are conducted and published which in fact do not have adequate power. Many years ago, this was a serious problem, as done by this review article in the New England Journal of Medicine, where in over a few years, 71 trials were examined, and in 50 of those 71, the authors noted that they could have missed a 50% treatment benefit. That would be a huge benefit in that the trials had very poor ability to find such major treatment effects. Today, it's probably less of a problem because of funding reviews and Food and Drug Administration reviews, but nevertheless, it's still an issue and it's still a problem that we cannot ignore. One of the first questions that the investigator may present to a statistician is, how many subjects do I need? Imagine in this example that we have a new treatment, which I label T, capital T, and we want to compare the benefit of that therapy to a control. Previous data from the literature or our own experience says that the control rate, the failure rate in the control treatment or the standard treatment is about 40%. I've symbolized that by P sub C. Now the investigator either hopes or believes that the new treatment could do better than that. In fact, it might be able to reduce the failure rate by 25%. That is, from a 40% failure rate down to a 30% failure rate, a relative 25% reduction, an absolute difference of 10%. So in that context, the question is, how many subjects do I need in each of my treatment arms? First is to recognize that the statistician can only give you a rough estimate, an approximate estimate, because of several uncertainties. The assumptions that we base the sample size on are not precise. They're based on the best available data, but certainly have some variability about them. So there's some uncertainty in our assumptions. Second of all, we usually are over-optimistic about the treatment effect. We have hopes, of course, that it's very effective, but nevertheless, the treatment may not be quite as effective as we hope. A third phenomenon is that patients who actually agree to be consented and randomized into a trial tend to be healthier than those that we have envisioned in our protocol design. We call that the healthy screening effect. That is, the event rate or the failure rate in the people we actually study in the control arm is usually less than what we assumed. So we need a series of estimates, not a single estimate, and we try to vary the assumptions and do the calculations and pick the sample size that seems most reasonable, the most stable. Clearly, one can be very conservative, so conservative that you price yourself out of the study. You can't afford it. So we need to be conservative and yet be reasonable. Well, how do we go about making these calculations? I'm going to introduce very briefly some statistical concepts which we really need to address. This lecture will try to avoid as much technical material as we possibly can. I'll only introduce what I think is absolutely necessary. So as you are aware, in science, we operate under the principle of the null hypothesis, that there's no difference until proven otherwise. So the null hypothesis, H sub naught, says there's no difference in the response rates, the failure rates, between treatment groups and control groups. However, we want to reject that idea and say that there is some difference. And in order to do the sample size calculation, as you will see, the estimate that we come up with depends on what difference you're trying to detect. We call that the alternative hypothesis. In this case, it's the difference of a specified amount, which I've called capital delta, that exists between treatment and control. So we want to reject the null hypothesis in favor of an alternative, some difference greater than 0, but in order to do the sample size calculation, we have to specify what it is you think is important to detect, because that'll be our target. In this context, there's two kinds of issues we also have to keep in mind of what we call the significance level and the power. The significance level, which is labeled alpha, is also sometimes referred to by statisticians as the type 1 error, or probably easier to understand is the false positive rate in our experimental process. It's the probability of rejecting a null hypothesis given that it's really true. That is, it's rejecting and saying there is a difference when in fact there isn't. That's an error. That's what we call the type 1 error, or false positive rate. And we want to control that to some low level, which we'll look at in a minute. The second concept is another error, that is rejecting the null hypothesis given that it's not true. That is, there really is a difference and we don't think there is. We call that the type 2 error. The power is 1 minus that type 2 error, and it's the probability of finding something that's really there. So with the null hypothesis, the alternative hypothesis, the significance level, and power, we can begin to formulate our strategy for computing the sample size. I now want to address another concept, and that is the issue of the normal distribution. This is a nice bell-shaped curve. It has some very nice properties. You've probably heard about this in other contexts. For our purposes, we're not so interested that the data are normally distributed, but in fact the test statistics that we use to summarize our data have a normal distribution, and I'll come back to that in a minute. But as you can see here, I've drawn in the top curve axis from -3, -2, -1, 0, +1, +2, +3. So we might say a standardized scale. And that's the scale that we'll be thinking of mostly for our test statistic. It's been standardized to this simple scale. You can think of this, though, if you wanted to, for data, where the data might be centered around some mean value, mu, and on each side of that there's some variability, 1 standard deviation, 2 standard deviations, and so forth. And if the data are normally distributed, or reasonably so, you can, in fact, use the normal distribution to describe that data. However, that's not our purpose for the sample size calculation. So we will use this distribution to say if there is, under the null hypothesis of no difference, that is, the test statistic would be 0, what are the chances, under that assumption, that the test statistic might get to be as large as, for example, 2, +2, or -2? And I've shaded in those areas under the curve to indicate that probability in the positive side would be 2.5%, or 0.025, and 2.5% in the other direction. So we can quantify the variability in our test statistic under the assumption of no difference. And if it's very unusual, we'd say, well, maybe our assumption of no difference really isn't the correct one. Well, how do we use that distribution in our sample size calculation? In this slide, I've indicated a little more complicated. On the left side, I've put down the curve from before that we would use under the null hypothesis, mu sub naught. And now imagine that there is a difference, which is the curve to the right, centered around mu sub 1. So you notice that the dark shaded area is the tail distribution of 2.5% that was from before. That would be the probability of a type 1 error, given that there is no difference, or the mean really is centered at mu naught. And then the cross-hatched area is the probability that would be less than some critical value, which we'll define, given that the mean really is at mu 1, not mu 0. Well, that would be the type 2 error, and the 1 minus that is the power. So we really are going to use this normal distribution to assess the behavior of our test statistic in evaluating two possible scenarios. One, that the null hypothesis of no difference is true, and another that some alternative is true. I should also point out that many of our test statistics, in fact, have this property of being distributed as a normal distribution with mean 0 and standard deviation 1. So here are some possibilities. We talk about a two-sided hypothesis or a one-sided hypothesis. In general, I favor the two-sided hypothesis, which says, under the null hypothesis, that there's no difference, that they're equal, and the alternative is that there's a difference in either direction. The one-sided hypothesis says, I'm only interested in a difference in one direction. We're not going to spend much time on the one-sided hypothesis because I don't think it's the proper or the best way to think about this. But nevertheless, it does exist as a formal way of testing your data. So in the classic test, then, we are going to look at the absolute value of our test statistic and ask, is it bigger, in the absolute sense, than some critical value, which we've called z sub alpha? What's this z sub alpha? That's the value we will use to determine whether the test statistic is unusually high or unusually low, given that the null hypothesis of no difference is true. So if you want to take a 5% type 1 error or false positive rate, then that critical value, under the assumptions I've summarized, would be 1.96, or roughly 2 standard errors of difference in that test statistic. If that were true, we would reject the hypothesis of no difference and say there is some difference, whatever it be bigger than 0. The one-sided hypothesis, which I said is not a favorite of mine, says I would reject the hypothesis of no difference, or that it's in one direction, if this test statistic is larger than the critical value, and if I want the type 1 error to be 0.05, or the false positive rate to be 0.05, the critical value is different. It's not the same. It's 1.645. You might say a less robust criteria for rejecting the null hypothesis. But at any rate, for our purposes, we're going to think about the two-sided testing hypothesis. And if you convince yourself, for some reason or other, that a one-sided hypothesis is demanded by the science or the ethics of it, then my recommendation is that if you use a one-sided tested hypothesis, it be tested at the 0.025 false positive rate, which in fact leads to the same critical value of 1.96 in either case. As we demand the same level of evidence for benefit, whether you view this as a one-sided or a two-sided case. This can be controversial, and you need to keep this clear. But in general, we recommend a two-sided tested hypothesis, at least at the 0.05 or better false positive error rate. So I've listed here some of the typical design assumptions in sample size calculation. 5% is the most common. It's the one you read about in most of the literature. If you want to have a more demanding criteria and take less of a chance of a false positive, then alternatives are 0.025 or even 0.01. The power is an important consideration, and you should design a trial that has at least a reasonable chance of finding what you're looking for. Conventional designs probably use a 90% power, the probability of finding what you're looking for to be 0.90 or greater. You should rarely design a trial that has less than 80% chances of finding what you're looking for, because then you begin to take a great chance of wasting resources, wasting patient efforts and contributions, and funding. So we're going to set our power calculations at least 0.80 and more likely 0.9 or better if we can afford it. The third very critical concept is to define this delta. It should be thought of as the smallest difference that you hope to detect, or the smallest difference that would be clinically meaningful. So in my little example, we talked about a difference in the failure rate between control and treatment from 40% to 30%, an absolute difference of 0.10, or a relative reduction of 25%. In this example, I have already specified what the question is. It's a failure rate question. Is the failure rate under control the same as the failure rate on treatment? Other questions would have different test statistics, but the concept of defining the difference is still the same, or at least similar. I've summarized some of the design assumptions for your convenience. If we're going to have a 5% type 1 error, the critical value, the Z alpha value, is 1.96. For 80% power, it would be a coefficient of 0.84. We'll come back and see how that's utilized. For a 2.5% significance level, it would be 2.24 for the critical value. For a 1% significance level, it would be 2.58. Notice as the level of the type 1 error gets smaller, or the false positive rate decreases, the critical value increases as you demand larger and larger evidence, stronger evidence to claim differences. On the power side, as we move from 0.80 power to 90% power to 95% power, notice that the corresponding coefficient changes from 0.84 to 1.645. That is, the power criteria also increases a parameter, Z sub beta, and you'll see how these work to determine the sample size. So the question is, how many do I need? Well, before you can answer that question, as I've alluded to, is what's the question? The sample size depends very critically on the outcome being measured and the method of analysis you're going to be using. When you ask the statistician, how many do I need? The statistician has to figure out what parameter you're going to measure, what variable, and how that variable might be analyzed, because that's the ultimate tool that will be used to detect the difference. So you have to think ahead when you ask the question, what's the sample size, or how many do I need? Here's a case to follow up the simple example, as I want to compare two failure rates. The null hypothesis, as I indicated, is that the control failure rate is the same as the treatment group failure rate. I can imagine, if I had such data, that I'd make that comparison by comparing the proportions observed, the failure rate proportions in each group, and use the normal approximation for comparing two binomials. I've indicated the formula there. You've perhaps seen it before in other situations or in courses you might have taken previous to this one. So it's simply the difference in the failure rates divided by the standard error of that failure rate. That's what's in the denominator. And the p-bar is the average failure rate in the two arms combined. I'm also going to assume, for the sake of convenience, it's not necessary, but for today's lecture, that we intend to have the same size number of patients in each arm of our treatment. As the number of patients in the treatment be the number of patients in control, which I'll summarize as being capital N. And I have specified, as I've already done, the delta between the failure rates in control and treatment. Given that I would use this test statistic, the sample size formula that I would come up with is as expressed in the formula on the top of the slide. It's not important that you understand how that formula is derived, except to remember that it is based on the test statistic that you intend to use to analyze your data. So different test statistics would have formulas that have the same form in general, but the details would look a little bit different, and we'll see some examples of that. So let's examine this formula just a little bit. In the numerator, you see two constants, two variables labeled z sub alpha and z sub beta. The z sub alpha, remember, is the constant that's associated with alpha or our type 1 error or the false positive rate. We might set that false positive rate, for example, to 0.05, and z sub alpha would be 1.96. The z sub beta is the constant that's associated with our beta, our power. So if we want to have 90% power, we would observe that the coefficient would be 1.28. We will use these coefficients then to plug into that formula to help determine our sample size. I want to point out that as the false positive rate decreases, the z alpha increases, and you can see that means that the sample size will increase. That is, z alpha plus z beta, the quantity squared, drives the size of n. So as you demand lower and lower false positive rates, it will increase your sample size. It's costing you. Likewise, if you want better and better power, that is 80% power, 90% power, 95% power, that coefficient also increases, which drives the sample size higher. So as you want lower false positive rate and higher power, those two parameters work together to increase your sample size. Notice the other two components. In the denominator is delta squared. Delta now is the difference in our failure rate between treatment and control. So as delta gets smaller, the difference that we want to detect gets smaller, the sample size will increase. If it was 0, you want to detect no difference. Of course, that's an impossible study. P-bar times 1 minus P-bar is essentially the variance of the parameter that we are looking at. So think of that term as variance, and as variability increases in the measurement that we're going to use, the sample size increases. Let's look at a specific example, this one we've been tracking here. We had the null hypothesis of no difference, PC is equal to PT. Our alternative hypothesis is that the control root failure rate is 40%, and we can reduce that from 40% to 30%. That delta in absolute terms is 0.10. Let's assume, which is commonly done, that we want a two-sided 0.05 false positive rate. The coefficient would be, for Z alpha would be 1.96. We want 90% power, which means that the coefficient for power would be 1.282. And the average failure rate between the two arms, 0.4 plus 0.3 over 2 is 0.35. With those pieces, we can now determine the sample size for this little simple example. Plugging those terms into that formula on the previous slide, we have 1.96, the type 1 coefficient, plus 1.282, the power coefficient, summed together, squared. We have the average failure rate, 0.35 times 1 minus that, or 0.65 in the variance term. And in the denominator, we have 0.4 minus 0.3, that quantity squared. Doing that calculation determines that the sample size in each arm would be 478 patients, or a total of 956 patients in our study. You may say that's an awfully large study, but we're looking for a relatively small difference, 0.4 to 0.3. This simple example illustrates the basic steps in almost all sample size calculations that we will be addressing in the next several minutes. So if you understand the basic steps here in different settings, we'll be repeating them again. I've shown you a table here which actually comes out of the text by Friedman, Furberg, and DeMets. And what's highlighted is the example we just did. The control group failure rate is 0.40. We want to reduce it from 0.40 to 0.30. I've given two options. In fact, I've given several options here. One-sided versus two-sided, 90% power, 80% power. So for the case we looked at, two-sided alpha, 0.05, and 90% power, you see I've highlighted 960. I've rounded numbers here generously. But now let's take this example a minute and say, well, 960 patients is probably more than I can do. 90% power would be desirable, but maybe I just can't afford that. Suppose I now decide that I can't afford 960 patients, that I would like to reduce the power from 90% to 80%, recognizing that I'm taking a chance of missing something that's important. You notice from this table that the sample size is not 960 any longer, but 720. Aha, we are moving in the right direction. We have a smaller sample size, but we've taken a chance, a greater risk of missing that difference of 0.10, and it's now not 10% chance of missing it, but a 20% chance of missing it. You might, although I do not recommend, convince yourself that this is really a one-sided hypothesis testing situation. For 90% power, the answer is 780 patients, and for 80% power, it's 560. Now, 560 might seem attractive to you, but remember, it's a 20% chance of making a false negative or missing the difference you're looking for, and it's in one direction. As suppose you're wrong and the failure rate is in the other direction, you'd have a problem. So while 560 might seem attractive from a statistical design point of view, it has some serious drawbacks. So despite the attractiveness of smaller sample size, we recommend a strong design with a two-sided, at least 0.05 type 1 error, and 90%, if you can possibly manage it, no less than 80% power. I've also graphed here to illustrate the point of how fast these things, sample size, can change depending on your assumptions. On the horizontal axis, I've given the relative reduction in the event rate from 10%, 20%, 30%, 40%, 50%. On the y-axis is the total sample size. And the curves represent the change in sample size for various event rates as the treatment effect changes. The dashed line, let's just focus on that for the moment, where it's labeled PC equals 0.4. That's the example we've been working on. And if we want a 25% reduction, which is the bottom line, we would notice that we'd get a sample size just under 1,000 patients, to be more exact, 960. But now suppose your assumptions were wrong. I've shown a dramatic change from 0.4 to 0.2 in the failure rate. That is, the control group isn't as sick as you thought and only fails at 20%, not 40%. Well, to keep your study well-powered, the sample size will no longer be 960, but something close to 2,500 patients. So your design is quite sensitive, or can be quite sensitive, to your assumptions. In this case, the failure rate is a key assumption. For that reason, we would be not wise to just do one calculation, but we should examine the sensitivity of our sample size calculations based on the assumptions we're going to make about event rates or failure rates in the control arm. The second, and you can see as the percent reduction in the event rate or the treatment effect changes, the sample size will also change. If we were to move from 25% reduction to 20% reduction, and imagine you drew the parallel curves, you can see that the sample size would climb even more. So this graph is a handy way to remind yourself of the sensitivity of the sample size calculation to the assumptions that you're making. In this case, the event rate in the control arm and the percent reduction or the treatment effect that you're trying to detect. Now, not all questions you will ask will be a failure rate. Some questions will be built around an outcome variable which is continuous. Variables such as blood pressure or some serum chemistry value, pulmonary function, and the like. The hypothesis that we're going to test might be comparing the mean values between the groups or the comparison of mean changes. So if that were the sample size calculation that we are going to address ourselves, how would that proceed? Well, we still have a null hypothesis of no difference. In this case, it's no difference in mean values, which I've labeled mu sub c and mu sub t, indicating the mean values in the control and treatment arm, or that the difference in those parameters, mu c minus mu sub t, is 0. As before, we have some idea of the difference we'd like to detect, which I've called capital delta. which in this case is a difference in the true mean values, unknown to us, but that was the hypothesis we want to test. If this was the setting, the test statistic that I might use to test whether those sample means are equal or not is indicated in the formula in the middle of the slide. X bar C minus X bar T, where X bar represents the mean value of the observations we make on the number of subjects we've examined. In this case, N sub C in control and N sub T in treatment. As I indicated before, it turns out conveniently that this test statistic also has a normal distribution with mean 0 and standard deviation 1, that classic bell-shaped normal 0-1 curve. It has nothing to do with the distribution of the data. It has to do with the distribution of the test statistic. So suppose, as before, that we let the sample size in each arm be the same, size n. I've indicated the sample size formula that would correspond to comparing two mean values. Notice the similarity of the form of this sample size formula. There's a z alpha plus z beta, as before, summed and squared. There's a delta squared in the bottom, as before. And there's a sigma squared, which represents the variance. So you can represent it as I have in the first term on the right-hand side of the equal sign, or you can do some algebraic maneuvering and represent the difference as how many standard deviation units, as delta divided by sigma. Either way is fine. It just depends on how you prefer to express the difference in your treatment arms. In either case, as z alpha gets larger, that is the type 1 error gets smaller. As z beta gets bigger, that is the power gets larger, the sample size will increase. As the delta, the difference that you're detecting, gets smaller, the sample size will increase. Notice it's squared, so it really moves quickly. And finally, the sigma squared represents the variability of the measurement that you are making. So these factors play off of each other to determine the sample size, and as I indicated in the previous graph, can be somewhat sensitive. I've also indicated that you can solve the equation in a different direction, saying if I have a certain number of subjects, a certain delta that I'm trying to get at to detect, and the sigma that goes along with that outcome variable, and I've pre-specified the type 1 error, I can calculate what the power or sensitivity of my study will be. We're not going to examine that in this lecture, but just let you know that you can calculate the sample size for a given difference, or for a given difference you can calculate and sample size you can calculate the power, and depending on your needs, either of these may be quite useful. So let me take a little simple example to test out how we would compute sample size for continuous outcomes. Suppose I'm measuring IQ. I've set the standard deviation for that measurement to be 15, and I have some memory improvement technique that I believe would detect the absolute difference of 4.5 in IQ, 3% of the standard deviation. I want to set my type 1 error to be 0.05, two-sided. I want my power to be 90%, and so I've specified my alternative as 0.3 sigma, or in this case 0.3. The sample size then is indicated in the formula. My type 1 error coefficient 1.96, my power coefficient of 1.282. I sum those, I square them. I have a difference of 0.3 squared in the relative difference in standard deviation units. Do the calculations, and it says my sample size is per arm is 234, or a total sample size of 468. You might indicate, well, that's too large. I can't possibly get that many patients in the time or the money that I have available. Well, then you have to go back and re-examine. Can I get a better measurement with a smaller standard deviation? Or is a larger difference perhaps acceptable? But that's a decision one must make cautiously, because you may still view a 0.3 difference to be clinically important. So one can lead yourself down a pathway that's not desirable in order to reduce your sample size. But nevertheless, it's a consideration you must make. Probably you don't want to reduce the power below 80%, but there are choices to be made, and so these must be done, and I recommend examined by building a table of various power parameters and differences of interest, and then choose what's doable, but also ethically and scientifically rational. Again, I've shown on this graph the sensitivity of your sample size calculations based on the difference you're looking for and the power that you're asking. So in this little case of a 25-30% reduction, you can see that for the power of 0.90, the sample size is close to 500. If you want to drop it to 80% power, you can see the sample size would be closer to 400. But again, these are choices you must make. there is no free lunch. Now, a third very common outcome is something called time to event. The primary outcome that you're going to use to assess your treatment is time to some failure, such as death, hospitalization, clinical events such as a stroke or a heart attack. And what you want to compare in this study is the distributions, or the survival distributions for the two groups. There are various measures one can use. One is what we call the hazard rate in the two groups, or it's similar to the ratio of the median survival time. We also have to keep in mind the length of follow-up in this study. How long will we follow the patients to allow them to have an event that were of interest? This setting is more complicated than the previous two. Here we have to imagine a model, and a common model is the exponential distribution, which models, in many cases, survival or time to event curves quite well. If we assume that we want to figure out the probability that our survival time exceeds some value, little t, we can model that by the exponential distribution of e to the minus lambda t. T is time, and lambda is the hazard rate. In our experiment, we're going to have two hazard rates, one in our group 1 or our control arm, and another in the treatment arm, say lambda sub 2. Under the null hypothesis, as before, we're assuming there's no difference. In this case, no difference in the hazard rates, lambda 1 and lambda 2, or the ratio is equal to 1. The alternative is we hope that there is a difference. In fact, we would like to see that the difference be improved in survival so that the hazard rate is one less than the other. So we can define the effect of our treatment by the difference in these hazard ratios and try to design our trial around that concept of change in the hazard rate. So if I follow this example in the simplest case, an exponential model, it will model survival, S of t, survival time is equal to e to minus lambda t, with lambda as the hazard rate. I can estimate the sample size by the formula labeled George and Dezou in the middle of the slide. Again, you will see the term z alpha plus z beta squared. Same terms as before, they influence the sample size in the same way. What's different here is in the relative way of expressing the treatment effect, and that is indicated by the log of lambda c to the log to lambda sub t, and that quantity squared. Again, that's the treatment difference expressed in this concept, in this term squared. This simple formula makes one very important assumption, is that we follow all patients until they have the event. We say that's no censoring, that we follow all patients till death, or all patients till they have a tumor recurrence, or all patients till they have a heart attack. That's not a practical assumption in the trials that we usually would do because of time and financial considerations. It also makes another very important assumption, is we get all patients immediately. As we have the pool of patients immediately available to us, it's a matter of how fast can we randomize them. Both assumptions are not realistic, but the sample size formula gives us a sense of how to start thinking about this. In this case, if we were to follow it through, the number of observations, number of events we would need is expressed by this formula, which is testing the formula that the ratio is lambda sub c to lambda sub I or lambda sub t. We can compute the required number of events that we need to see, and I won't go through the details here, but simply to say the number of events you expect to see in a trial, to the D, is how large a population you start with, and what's the probability of an event. The probability of an event is a function of how fast you recruit patients, how long you follow them, and what the average hazard rate is, and the bottom bullet is a formula which summarizes all those factors. As I indicated, the simple formula makes two very important assumptions: that their patients are all followed to the event, there's no censoring, and that you got patients recruited at the same time. Well, suppose we try to relax those assumptions and say, well, we can't possibly follow all patients to the end, but we will have some what we call censoring, a point at which we don't know whether they had the event or not. And we will do our best job, but patients will be recruited over some period of time, or what's usually referred to as staggered entry. The formula at the bottom of this slide, under the label of the leukine, is a model which has a very general form, but it's of the same form. That is, there's a z sub alpha plus z sub beta, the quantity squared, in the numerator. In the denominator is the difference in the hazard rates, lambda sub c minus lambda sub I for intervention, and that quantity squared. And then there are these g functions, G of lambda C and G of lambda sub I. These terms represent the variability of our observed hazard rate depending on these factors of how fast do we recruit and how long do we follow. So I've indicated here three formulas that get more and more complicated. The first one assumes that we're somehow able to recruit all patients immediately and they get censored at some time, capital T. The second formula is more relaxed. It says, well, we won't get them recruited all in some time frame from 0 to capital T. We'll recruit all along, but at capital T, we stop the trial and analyze the data. The third formula is more general, and it says we're going to do our best job to recruit all our patients in the interval from 0 to T sub 0, and then we'll follow them for a little while longer to capital T. So we're going to recruit for a while and follow for a bit longer. And this is probably the most common setting that we would use in typical clinical trials. So you can see from case 1 to case 2 to case 3, the formula becomes more complicated because it's reflecting the ability of our precision of our estimate of the lambda. The less precision, more variability, larger the sample size. I've given an example just to indicate how this might work. Suppose, again, we want to have a trial that's designed with type 1 error of 0.05 and a power of 0.90. From previous data or previous trials, we know that the hazard rate and failure rate in the control arm is 0.3. We want to reduce it from 0.3 to 0.2. We think we can get our patients followed for a maximum of 5 years. That's the length of our grant, for example. And we know that we'd like to get the patients recruited as fast as possible, but the best we can do is probably keep recruitment over for 3 years. Using these 4 models actually, the first case is the unrealistic assumptions of following patients so they all have an event and instant recruiting. But that was a sample size in one arm of 128 patients. If we recruit instantly but follow patients to capital T, which is 5 years, we'd have more patients in the study, 188. Why? Well, we don't have as many events. We're not following all patients to the end. So we have less data, less precision, need more patients. Suppose we censor at capital T 5 years and we recruit all the way up to 5 years. So we need 310 patients. Why? Well, not all patients are available immediately to be followed. They're not at risk for an event. So less exposure, less risk, less events, need more patients. Suppose we say, well, we can probably do better than that. We can recruit in 3 years, T sub naught being equal to 3, and follow for 5 years. What happens then? Well, you're getting patients in faster. You're going to quit after 5 years. So you should need fewer patients because more patients are being exposed than in the previous case. Well, the answer is yes, you do need fewer patients, 233. What I wanted to illustrate with a simple example is that the sample size will change depending on how you design your trial for follow-up time, recruitment time, type 1 error, power, and your estimates of the failure rate in the control arm. There's another factor that must be taken into consideration when you are designing a trial. Because we're dealing with human subjects, not all patients will adhere perfectly to the treatment protocol. They may take less of the therapy. They may decide not to be followed for as long. So we call this non-adherence. The impact of non-adherence is it will dilute whatever treatment effect exists, the true effect. Whether it be beneficial or harmful, non-compliance will dilute that effect. If you don't take that into consideration, your trial inadvertently will be underpowered. You will not have the ability to detect the differences you're hoping for because the effects are diluted. There's a very fundamental principle which we'll talk about in a couple of more lectures, which is called the intention to treat principle. This is very important because what it says is that you must account for all patients that you randomize in your analysis. If you drop out patients out of the analysis who didn't comply, you can introduce substantial bias. So we can't compensate for that kind of bias in the analysis. The alternative is to try to compensate for that in the sample size calculation by adjusting to compensate for this dilution effect. We talk sometimes about 2 kinds of non-adherence. The dropout is a patient who was assigned to the treatment arm, even if it's in a blinded fashion, and stops taking the study medication. So they're not getting the benefit of the therapy that they might have. The drop-in patient, less common but nevertheless still a factor, is that the patient in the control arm may actually, through alternative means, be able to start taking experimental therapy. For example, today, if you were to test a trial with 2 doses of aspirin, well, the patient may not know which aspirin arm they're in, but they may decide on their own to take a higher dose of aspirin because it's available. They can go down to their local drugstore and get it. That'd be an example of a drop-in. Dropouts and drop-ins both have the same effect. They will dilute the treatment effect that you're trying to test for. So I have a little graph here that illustrates the impact. On the continuum of 0 to 1, this is failure rates just to make it simple. Imagine we have some large failure rate P sub C, and we want to reduce it all the way to the left to P sub T. That would be a major reduction. But we have both dropout and drop-in going on. What's the impact of that? For the treatment arm, instead of having the benefit of treatment, which would reduce it to P sub T, we have less benefit. So you move up in the failure rate from P sub T to P sub T star as a result of dropping out. On the right-hand side, we have the control rate of P sub C, and if the patient starts dropping in, i.e. taking the therapy because it's available elsewhere, they get some of the benefit. So the rate is not as high as P sub C, it's a little bit less, P sub C star. The important thing to notice is that the difference between P sub C and P sub T is not what you expected, it's less, indicated by the 2 internal arrows. Well, that's the impact of the dilution effect, and therefore the delta that you're going to be using really in your study is less, and therefore if you think back to the sample size calculation, you either need more subjects or accept less power. So I've indicated a very simple little formula here to say what would the sample size adjustment be. Suppose we don't have any drop-ins. Let's just worry about one factor, the dropout, the patient who does not take the study therapy and they're on the treatment arm. Well, in that case, the control rate should be the same because nobody is dropping into the therapy. But the treatment effect, failure rate, will be made up of 2 pieces. The part that you would hope for, the 1 minus R, the percent of effect times the treatment failure rate, plus those proportions R who quit on you times the original failure rate. So you have those who are getting the benefit of the therapy, that's the first term, plus those who are not getting the benefit of the therapy. Those 2 together give you an average event rate in the treatment arm. If you do some algebra, you can show that the adjusted sample size for that dropout phenomena would be the original sample size modified by 1 minus R squared in the denominator. If you examine this impact, suppose you had a 10% dropout rate. Might be quite reasonable for a typical study. The inflation factor is 1 over 1 minus R, the quantity squared. In this example, 1.23. That means in order to compensate for a 10% dropout rate, you would need to increase the sample size by 23%. Suppose things are a little bit worse. You have a 25% drop-in rate. That means in order to compensate for that dilution effect, you'd have to have a 78% increase in sample size. This concept is extremely important because it reflects back not only on the sample size calculation, it reflects on what you must do in your study to assure or to maximize compliance. You can't analyze your way out of it. You must build it into the design. If you don't build it into the design up front, if you have non-compliance at a higher rate in your conduct than expected, you will be stuck with a trial with decreased power, and there's no fix at that point. I've now added the drop-in component as well as the drop-out component indicated by R sub 0 and R sub 1. And you can see in the graph, in the formula below, that they work together in the same fashion. And then below I've given 2 examples of a 10% dropout and a 10% drop-in. That would imply a 56% increase in sample size, quite realistic in many trials. Suppose things are a little bit worse, 25% dropout, 25% drop-in. That would require a 4-fold increase in sample size. So the non-adherence is serious business, and it must be considered in the sample size and the design of your trial. There are many more complicated models. We will not discuss them here in detail, but just say one can model it even further by looking at the length of follow-up. The hazard rate may vary over time. The non-compliance rate, both dropout and drop-in, may vary over time. And the treatment effect itself may lag. If you're looking at cholesterol lowering, the effect may not be immediate. It may take several months or even years for the effect to take place. So one can model all of this, and they do have impact on the sample size. Give you one example using this more complicated model. a trial called the beta blocker heart attack trial using a beta blocker in patients who had survived a heart attack. Two-sided 0.05, significance level, 90% power, 3 years of follow-up in the trial. The failure rate in the control arm, based on previous trials and data, was expected to be 18% over this period of time. It was hoped that the treatment effect would be 28%, 18% to 13%, and the dropout and drop-in rates are as indicated, which varied over time. Dropout was estimated to be 12% in the first year, 8% in the second, 6% in the third. Drop-in was somewhat unknown, but it said 6 or 7% per year was estimated. If you put all those pieces into this more complicated formula, which we've not looked at in great detail, the result is that the event rates are adjusted because of this dilution, because of the varying factors. But what's important to recognize is instead of getting a 28% reduction as assumed, you really are detecting or have a trial that's designed for a 20% reduction. And the sample size goes from a total of 2,200 to a sample size of 4,000. So these factors are important. They have serious effect, and you will not always know precisely all those factors, but you must build yourself a table that indicates if I vary this factor by a little bit, what's the impact of my sample size, and making a judgment at the end of the day as to what sample size is realistic within your time frame and your budget. A challenge which is really in a lot of discussion these days is the equivalency, or better termed, the non-inferiority trial. You want to compare a new therapy with the standard, and you'd like to really show that the new standard is just about as good as the old one, but it has advantages. It may be less costly, less toxicity, or maybe it's profitable for your company. There are many examples of trials which have been done. One is the intermittent positive pressure breathing trial, which compared an expensive procedure, which was called IPPB, versus a cheaper treatment. The nocturnal oxygen therapy trial compared 12 hours of oxygen therapy in patients with chronic obstructive lung disease to 24 hours. Obviously there, difference of cost, difference of convenience. The problem, however, is you can't show no difference. If you look back to the sample size formula we just covered, assuming a delta of 0 gives you an impossible study. So while I've oversimplified this problem greatly for the purposes of our discussion, the solution is that you must specify some minimum difference that's of clinical importance that you'd be willing to give up in treatment effect in order to take advantage of the other factors of cost, invasiveness, toxicity, and so forth. This is very challenging. And again, as I present the sample size formula, you can see that if you can specify the minimum difference that you're interested in and make the other assumptions, you can get a sample size that will give you a pretty good idea of what you would need to do a non-inferiority study. The exact details of this are much more complicated than we've covered here, but at least it will give you some sense of this problem. The challenge of specifying non-inferiority, though, is really a daunting one. I've shown here in this graph differences in failure rates. 0 difference would be that the null hypothesis is true. And what's implied here in this graph is that you'd be willing to give up a 2% increase in mortality, or failure rate, whatever it might be, for the sake of some other effect, some other benefit, less invasive, less costly, less toxic. So we've indicated a series of trials. So trial A, the top one, which is indicated the estimate of the difference of treatment effect, and the line indicates the plus or minus 2 standard errors. So the first trial is superior because it shows, in fact, you can rule out no difference. It says that the new drug is, in fact, better. But for non-inferiority, what you want to do is rule out that it's not worse than 2%. Well, trial B clearly meets that goal because the estimate is favorable to the new treatment. It doesn't rule out 0 because of the length of the constant interval, but it does rule out 2% quite clearly. So that would be pretty good. You'd be very satisfied with that trial if that were the results for a non-inferiority design. Trial C is a little bit more worrisome. The actual estimate is on the side that favors the standard by a little bit, but nevertheless, the confidence interval says that the effect is not worse than 2%. You still are non-inferior even though you're a little bit worse. Trial D shows that it's not inferior. The estimate is less than the difference of non-inferiority, but it is clearly worse than no difference. Trial E is even worse in that it is worse than no difference, and it's far worse than the zone of indifference. What you want to avoid is the last trial, which is so small, so imprecise it doesn't tell you anything. So part of the challenge of a design is to make sure that you have an adequately powered, adequately sample-sized trial so you don't wind up in case F. Now, there's lots of discussions these days about adaptive designs, or what you might think as midstream adjustments. As I said, Murphy's law applies to sample size. Even the best assumptions will not be exact. And you may find yourself in a situation where the assumptions are way off from the early results and that the power will be inadequate. What do you do in such a situation? Do you just quit? Say, well, that didn't work out? Or do you take the stubborn approach and say, I'm going to continue on and finish what I started no matter what, but realizing that there's certain doom that you'll be successful? Or can I adjust the sample size in some way that would allow me to rescue this design? Or perhaps I can extend follow-up if I'm looking at a time to an event study. In other words, can I make some mid-course corrections or adaptations? The rule of thumb in this is that you need to do these decisions early, and best not to look at treatment comparisons, although recent research has developed some methods that allow us to do that in a technical, statistical sense, but no one is quite sure yet how to implement those methods without opening the door for bias. So in general, one wants to make your mid-course corrections early. Try to make them without depending on the early treatment effect because it is unreliable and may lead to biases. I want to thank you for your attention for this very important subject. Next time, we will be examining monitoring of studies for early evidence of benefit or harm. In this lecture, we examine carefully the principles of sample size estimation, recognizing they are a function of many factors, including the false positive rate, the power, the event rates or response rates in the control arm, and the size of the difference you're trying to detect. Sometimes these factors can't always be determined with precision, but you need to examine the robustness and the sensitivity of your sample size calculation as these estimates may vary. Thank you very much for your attention.